Hi, I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce our last talk of the day, CRISPR-Cas9 editing on the Arginine Vasopressin v1a Receptor in Syrian hamsters, lessons learned and future directions. I'm pleased to welcome Jack Taylor as your presenter. Jack completed a bachelor's degree in psychology at Westminster College in Pennsylvania, where he studied the relationship between pain perception and athletic performance among college athletes. He then completed his master's and doctoral training with Dr. Jeff French at the University of Nebraska, Omaha, when he studied the roles of oxytocin and vasopressin in modulating social behavior within marmoset monkey families. While in Omaha, Dr. Taylor also characterized the binding and signaling properties of the oxytocin vasopressin 1a receptors in 4 different primate species, when bound to a novel form of oxytocin found only in South American monkeys. In 2018, he moved to Georgia State University to study the roles of oxytocin and vasopressin in modulating aggression, social communication, and social reward. A warm welcome to you, Jack. Following the presentation, we'll have a Q&A session and we'll welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Jack's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon on the menu on the left-hand side of the player. Now, without further ado, I will hand over to Jack to begin his presentation.
Thanks, Caroline. So today I'm very excited to be talking to you all about some of the work that I did at Georgia State University, where we generated the world's first v1a receptor knockout Syrian hamster. It was a long process and we learned a lot, and we have a lot of exciting ideas for the future, and I'm excited to tell you all about those. So before I begin, I want to acknowledge the phenomenal effort that this was amongst a lot of people. I want to call special attention here to Dr. James Walton and Dr. Kate McCann, who are my co-first authors on this paper. This really was the work of several labs and 3 postdocs who all shared equal work on this, and we sort of worked sequentially. So Kate was the one who started the project, James kept it going, and then I was lucky enough to finish it off. This paper has 4 senior authors, plus an attending veterinarian, along with all of the graduate students, a wonderful lab tech, and then, of course, the 3 postdocs who headed the project. So I wanted to show this image of the vasopressin network in the brain. And as you can see, there are several brain regions in the brain that produce or receive vasopressinergic input. And you can see that they are very well connected. And the reason that I wanted to show this is because neuroscience has gotten a long way with targeted ablation studies and pharmacological studies targeting single nuclei in the brain. And we've learned a lot from these methods, and we've gotten very, very far from these methods. But as genetic models advance and as just new models advance, we really have to start thinking about the brain as a network and how interconnected all of these brain regions are. And so as we look forward into the future of neuroscience, I really think that we need to be thinking about the vasopressin network and all brain networks as just that, networks. So I'm going to make a case that I think that we need new model species that are genetically modified. Now, we've gotten a long way with rats and mice and also the zebrafish that have traditionally been genetically modified and have produced a lot of very, very valuable genetic models. But I'm going to make the case that we need even more. We need more new rodent species in addition to mice and rats. And the reason that I say we need genetically modified animals is it is only with control of the genes that we can control the expression and the activation of a gene or a gene product. So we can administer all the receptor antagonists that we want, but we'll never completely block or remove the input of a receptor or a protein. We need to knock out the genes or at least turn them off. In many cases, genetically modified species are easier to use, especially when we're talking about knockout models. The manipulation in many cases is already done. All the researcher has to do is breed. And so this makes them very available to researchers and especially students as well that don't need to do surgeries, they don't need to do injections. The manipulation's already done. It's bred into the animals. Now, I said mice have been incredibly important for biomedical research and have taken us very, very far in neuroscience and in biomedical science. But there are some behaviors that mice just don't effectively model. And there are some diseases that mice just don't effectively model or are more effectively modeled in a different species that maybe expresses that behavior naturally. In addition, when we have the same behavior controlled by the same mechanism across species, these conserved mechanisms, we can be a lot more confident that when we translate that phenomenon from rodents to humans, that it's going to effectively translate, and then that mechanism is going to be conserved. If we just study a behavior or a mechanism in one species, we're less confident that that's going to translate to human health and behavior versus if we see the same mechanism in hamsters and mice and monkeys and fish. We'll just feel a lot more confident that that's going to translate and that that money will be well spent on the human research. Similarly, subtle differences in the way that mechanisms work between species can help us predict how that will translate to human health and behavior as well. So even if the same behavior doesn't work the same way in every species, the common mechanisms and the subtle differences can really tell us a lot. So hopefully I've convinced you that we need new model species, and I'm going to continue to try to convince you that we need my favorite non-mouse rodent, the Syrian hamster, and that they are a very effective model for a specific subset of behaviors, specifically social communication, aggression, and social stress. Now, the first behavior that I'm going to talk about today, and you will hear a lot about this behavior, is flank marking. And so this is a territorial scent marking behavior that all hamsters do starting at about 15 days of age. Males and females do this behavior. And essentially what it is it's very similar to a dog peeing on a tree. It's a territorial scent marking behavior. What they'll do is they have got these glands on their hips or on their flanks, and they will groom that, and they will mix it with their saliva, and then they'll rub their flanks against anything they can get access to. In our case, it's usually the edges of the cage. What's really great about flank marking is it's very easy to score. It's very robust. Male and female hamsters do it at similar rates, and it can be robustly stimulated in the laboratory either by exposing the hamsters to conspecific odor or with pharmacological agents. The next behavior that I think hamsters are really great at is to study same-sex aggression. Aggression in hamsters occurs in both males and females, male-male aggression, female-female aggression. And the reason it's so great for a laboratory study is it's highly stereotyped. And what this means is same-sex aggression is sort of like a dance. They'll sniff each other in a little circle, and then they'll kind of get in a couple little wrestling matches or tumble fights. And then very quickly, it is determined who the winner is and who the loser is based on who is stronger and who's weaker. And very quickly, the aggression is over. The loser will submit by raising his tail and running away. And the winner will win by chasing. Because it's so stereotyped, very rarely do injuries occur. This is good from an animal welfare standpoint, but this is also good because we don't want to study physical injury or infection. We want to study the social effects of aggression. Hamsters will do this readily. They are territorial and they are solitary living. And so we don't have to do anything that is unnatural to the hamsters to get them to do this behavior. With some other species, sometimes you have to stress them out to get them to be aggressive. You might have to use a different strain of the same species, or often a lactating dam will be used as an aggressor against a same-sized or a smaller male or female. And because they perform this aggression naturally, because it's stereotyped, because they can quickly figure out who the winner is and who the loser is, hamsters will develop stable hierarchies of an alpha, a beta, and even those more subordinate than that. Because they develop these stable hierarchies, they're also a very good model for social stress. Other humans are the primary form of stress for humans, and so it's really great to be able to model that very effectively in the laboratory in a rodent. Hamsters, after they are defeated, after they lose a match, they develop this defeated phenotype, and it can be produced after a single loss, and they will remember this phenotype. Essentially, they remember that they are a loser, that they've lost, and they will behave submissively not only to their original aggressor, but also to any new hamster that they come across. They will submit and exhibit this defeated phenotype. And so that makes them really, really useful for social memory and for the study of social stress. And hamsters are similar to humans in a lot of very important ways. First of all, cortisol is the primary stress glucocorticoid in both humans and in hamsters, whereas in mice and rats, corticosterone is the primary stress glucocorticoid. This may seem like a small point, but it's very important for stress research that we are studying the same glucocorticoid in both our rodent model and in humans. This information is a little bit dated at this point, but hamsters were able to contract the very first variant of COVID-19. It seems like these newer variants can infect mice as well. So this information is a little bit dated, but hamsters also develop cancers that are similar to humans. The way that they're vascularized, the way that they metastasize is very similar to the way that it does in humans, and so they can be very important for cancer research. Okay, so hopefully I have convinced you that hamsters are a great model for all these behaviors. I'm going to talk a little bit about flank marking because I think it's such a great biomarker for vasopressin in the brain. So like I said, flank marking is this behavior where they will lick and wet their flank glands. You can kind of see one in eye there, that little dark spot on this hamster's back. And then they'll rub it on whatever they can find. And in image number 2, it's rubbing it on the side of the cage, which is kind of tough to see because it's translucent. And what's great about this is it can be induced in the laboratory very robustly. So on the right-hand side, we've got flank marks on the y-axis, and on the x-axis, we've got hamsters that have been placed into the dirty cage of another hamster, that scent side, or a clean cage, that no scent. Don't worry about the LP and SP for right now. The point here is that when you place a hamster into a dirty cage or a cage that has been marked by another hamster, essentially all they do for the next 10 minutes is engage in this flank marking behavior, and they don't really do it in a clean cage. So what's also great about this is that this can be exhibited pharmacologically. So on the left-hand side, and on these two image videos are the same hamster, and one of them has been treated with saline, and the other one has been treated with a vasopressin agonist. And I'll let you all take a guess at which day this hamster was treated with a v1a agonist. Like I said, this behavior is really great because it's very easy to score. It's very robust. All of you, probably by now, are experts in flank marking, which means that it's very quick to train students to score this behavior, and we get very reliable estimates of how many of these instances of flank marking these animals exhibit. I should note that these are clean cages, so there's no existing scent there from another hamster. It's the same hamster. One day they're treated with saline, and the other day they're treated with v1a agonist. It should be pretty obvious by now, but I'm going to show at the bottom here. The hamster on the left was treated with a vasopressin 1a receptor agonist. And we know that this vasopressin is very, very important for eliciting flank marking behavior. So like I said, vasopressin is one way to induce this behavior in hamsters, and it's very, very robust. This occurs in both males and females. If you give a hamster arginine vasopressin or a v1a agonist in the brain, I think this one is intracerebroventricularly. That's all they do for the next 10 minutes is flank mark. It occurs in males and females at similar rates. It's somewhat specific to areas within the hypothalamus, specifically the MPOA and the anterior hypothalamus. You should note here that there is one other hormone that elicits this behavior, that is oxytocin. Oxytocin is very closely related to vasopressin. They only differ by 2 amino acids, but the response is stronger with vasopressin. This occurs in single-housed animals and in subordinates, so it's not as dependent on housing status or social status as, say, odor-stimulated flank marking is. And I said that oxytocin will elicit this behavior. The reason that oxytocin elicits flank marking is because oxytocin can bind to the vasopressin receptor. So when oxytocin is inducing flank marking, it's not because it's binding to the oxytocin receptor, it's because it's binding to the vasopressin v1a receptor. So we know that this behavior is specifically controlled by the v1a receptor. So vasopressin also helps control aggression in hamsters, and it's a little bit different than flank marking is. I'm going to specifically show a couple of bars in this graph. So on the y-axis, we've got the mean duration of aggression, and in the red box, I'm showing vehicle-treated animals. And the reason I'm showing this is to show that males and females will express aggression at pretty similar rates under normal conditions. But when you treat a male hamster with vasopressin, they become more aggressive, and when you treat a female hamster with vasopressin, they become less aggressive. So we've got this sort of opposite effect of vasopressin for aggression, whereas for flank marking, it increases in both males and females. So because we have this really deep wealth of knowledge about how the vasopressin 1a receptor controls both social communication behavior and aggression, about 15 years ago now, my mentor and co-author, Elliot Albers and Kim Human, they had this idea to develop a v1a receptor knockout in Syrian hamsters using CRISPR-Cas9. And so that was what was done. So this work was begun by Kate McCann. So what was done was a single guide RNA targeting the start codon of the v1a receptor was generated. So I've got it kind of color-coded here, that the guide RNA is in blue, and it's sort of, you can see the start codon there in the middle of it, and then all single guide RNAs, in order to work to interact with the genome and with Cas9, they need this protospacer adjacent motif, this PAM region. And so you have to pick your guide RNA based on where there's a PAM, but with software, it's relatively easy to do this. So then this guide RNA was injected into single cell embryos along with Cas9, hopefully to create a break in the v1a receptor gene, and then hopefully due to non-homologous end joining, inducing a frame shift mutation, either a deletion or an insertion that results in a disruption in the production of that gene. We targeted the start codon because we hoped that we could just disrupt the start codon and not have any gene product at all. but you're sort of at the whim of randomness without more sophisticated forms of Cas9. Once those animals were born, we ear punched them. We took those ear punches, we extracted the DNA, we amplified it, we cloned it in E. coli, and then we sequenced a number of clones from those colonies, and we looked for animals that had an insertion or a deletion. And then we took those mutants and we bred them, and then we bred them until we had a knockout. And I'll sort of explain why we didn't get a knockout right at the beginning. But you breed the mutants together to get enough animals that you can do some phenotyping. In our case, it's mostly behavioral phenotyping. So one thing that came from this is we found that embryo injection of Cas9 and this short guide RNA or single guide RNA very efficiently induced frame shift mutations. So there weren't that many injections actually done, and we got a lot of frame shift mutations from our animals. So you can see here in A, we've got a number of frame shift mutations. On the top, we've got the wild type gene, and then everything below is one of the mutants that we had. Only one of our animals was actually able to produce viable offspring. We call her C1, she's our founder, and she produced all of the offspring and the descendants of the offspring that will be phenotyped from here on out. She had, from her ear punch, we extracted a 47 base pair deletion from her, which was really great. It was great that we had this big deletion. It spanned the start codon. But what we found was that in her offspring, we didn't find this 47 base pair deletion. We found both a 10 and an 11 base pair deletion, and this persisted into generation F2 as we sort of got our genotyping protocol down. And what this tells us is, while we were able to extract and amplify a 47 base pair deletion from her ear punch, what this means is that in her germline cells in her eggs, she had a different mutation. She had these 10 base pair, these 11 base pair deletions. And what this means is that she was a mosaic. Our founder, C1, was a mosaic, that we injected Cas9 in our short guide RNA or single guide RNA at the single cell embryo stage with the hope that it would affect all of her cells, because you start at one and that divides from there. But what must have happened is that additional cuts and breaks happened after that single cell had divided. And so what that means is, in her ear, we found a 47 base pair deletion. In her germline cells, we found 10 and 11 base pair deletion, because we found those in her offspring. And we don't know what her other cells might have had, but she was a mosaic. Luckily, this 10 and 11 base pair deletion, they are both frame shift mutations, and while they contain the start codon, they result in a truncated protein. We think it probably looks like this. Because the start codon is conserved, we expect that it would start, that the start codon would pick up right where it's expected to, but what it leads to is a premature stop codon, as well as some other mutations from the wild type protein, which is shown at the top. It's nice and long, but our 11 base pair deletion produces this sort of truncated and incorrect gene product. Because Cas9 can induce off-target cutting. CRISPR tends to be very, very specific, but there is a possibility that our short guide RNA, our single guide RNA, can bind to and cut at other sites. And so we searched against the entire hamster genome, and we looked for places where they weren't great matches, but where there might be some match to other proteins. And we identified these proteins. We were only able to clone and amplify these two. It's very difficult. We didn't have a great hamster genome at the time, and it was difficult to develop PCR primers for these other genes. But this SCARF2 and this PROB1 both of them were identical to the wild type gene, and because we started with a mosaic, we outbred several generations before we started with any phenotyping, hopefully sort of diluting out any off-target genes and keeping only our intended knockout. Okay, so we had evidence that the knockout works on a genetic level, that it produces this truncated protein. We needed to make sure that it wasn't working at all levels. So the first place that we looked is in the brain. So we have a really great method for looking at v1a receptors in the brain through autoradiography. Here are just sort of a smattering of brain regions. So on the left, we've got wild types, WT. In the middle, we've got heterozygotes that have one copy of the CRISPR allele. And on the right, we've got knockouts, or KOs, who have both copies of the knockout allele. And you can see, you've heard of t-test. Well, this passes the I test, as in you just have to look at it to see that there's a difference between the wild types and the knockouts in the amount of v1a receptors that are available in these brain regions. I could show you all the other brain regions, but it's the same story. Now, we wanted to also look at the oxytocin receptor because we wanted to check two things. Number one, that the knockout didn't affect the oxytocin receptor in terms of downregulating expression through some sort of off-target effect. And we also wanted to check that the oxytocin receptor wasn't upregulated to sort of counteract the loss of the vasopressin receptor loss. And what we found is that in our wild types, our hets, and our knockouts, the oxytocin receptors were essentially unaffected in all of the same brain regions, as well as a few extras. So I can show you these bar graphs. They look great, and you can see there's an obvious difference. But really what is telling is looking at the actual images themselves. So what you're looking at here is some sections of brain from a wild type, a heterozygote, and a knockout. And anywhere you see dark development of the film is where there are dense amounts of vasopressin receptors. And you can see there are dense pockets of vasopressin receptors in our wild types and in our heterozygotes, but you can barely even see the outline of the brain section for the knockout. And I promise you, it looked this way across the entire film. You can tell which sections were knockouts from about 3 feet away because they're just not there. One additional thing I should mention is you can't tell from this photomicrograph, but in several important brain regions, heterozygotes were sort of intermediate between wild types and knockouts, which suggests that having one copy of the knockout allele and one good copy isn't quite the same as having 2 good copies of the gene, something we call haploinsufficiency. So we showed that the v1a receptors are not present in the brain, but we wanted to also show that in some functional ways. So here what we've got is, so the v1a receptor is present in the brain, but it's also present in the periphery, where it's very important for the baroreceptor reflex. Essentially, it controls blood pressure. When you give a selective v1a agonist, here we used v2-OVT, it very quickly increases systolic blood pressure in anesthetized wild-type animals. But when you give the same drug to an anesthetized knockout, there is no increase in blood pressure, suggesting that while there are no v1a receptors in the brain, and also there are no functioning v1a receptors in the periphery to produce this blood pressure response to a vasopressin agonist. Okay, so we showed biologically that there is no effect. We also wanted to show behaviorally that the v1a receptor was knocked out. So you remember from our video earlier, if you give a hamster, male or female, v1a receptor agonist, and you put them into a clean cage, they flank mark. That's what that video on the left was. So what we did was we gave a v1a receptor agonist to our knockouts, and then we put them into a clean cage, and we did the same thing with wild types and heterozygotes. And you can see when on the left there, our wild types, when they're given saline and put into a clean cage, they don't flank mark. But when you give them AVP or a v1a agonist, they spend the next 10 minutes essentially flank marking every inch of the cage. Our knockouts, however, when they're put into a clean cage, they don't flank mark, whether they're given saline, vasopressin, or a selective v1a agonist. So what this shows is there's a functional loss of vasopressin-induced flank marking in our knockouts. So this is the most important experiment here. We wanted to show that we expected that we would have a loss of flank marking in odor-marked cages. So if you remember, if you put a hamster into a cage, a dirty cage that's been marked by another hamster, they will spend all of their time flank marking. So we put our knockouts, our wild types, and our heterozygotes into dirty cages, and we observe their behavior. And contrary to our expectation, our knockouts flank marked more, almost twice as much more than their wild type litter mates. And this is very surprising because we expected, we had all these years of research saying that vasopressin induces flank marking, that vasopressin alone can induce flank marking. And so we expected if we'd knock out the v1a receptor, we'd lose odor stimulated flank marking, when in fact, they actually flank mark more than their litter mates. This is a very surprising result, and so we repeated it in a slightly more controlled way, in a slightly more efficient way, and also in a totally separate experiment with a separate experimenter. So we put hamsters into clean cages and we put them into odor-marked cages. And what we found is that this effect was replicated, and it's specific to odor-marked cages. So it's not that they're just highly reactive and they'll flank mark anything. It's specifically induced by odors. Again, our wild types flank mark a lot, but our knockouts flank mark twice as much or more in our odor-marked cages. And this is an important replication and also showing that it is specific to odor-induced flank marking and not just novelty. So if you remember a few slides ago, I said that vasopressin makes males more aggressive and makes females less aggressive. So we expected if we knock out the v1a receptor, we would expect females to become more aggressive and males to become less aggressive. And so what we did is we placed our knockouts, wild types, and heterozygotes into the cage of a smaller same-sex hamster, essentially one that they can choose whether they will be aggressive or not, and that smaller one won't be aggressive towards them. And unexpectedly, we found that there was no sex difference in aggression between our males and our females of our knockouts, and that vasopressin knockout increased aggression over their wild type litter mates. And so this was also surprising because we had expected sort of this sex difference and we expected males to be less aggressive, but in fact, they were more aggressive. And so we were initially, kind of flummoxed about this. We didn't know what to make of it. We had these expectations. We thought we knock out this gene, we're going to get no flank marking, and it'll be one and done experiment. It'll be very easy. And we got these unexpected results, but maybe they shouldn't have been so expected because we have some evidence from photoperiod research that kind of suggests that maybe v1a receptors aren't the whole story for social communication and aggression in hamsters. So fun fact about hamsters, they are extremely sensitive to photoperiods. When they're housed in winter-like conditions where they've got more darkness during the day than lightness, they've got a shorter photoperiod, they go into this winter-like phenotype where they become less aggressive, they put on weight, and their males, their testes actually regress and become smaller. When they're housed in a summer-like photoperiod where they've got more light during the day, they become stronger, they become more aggressive, and their testes grow in weight. And this, but this is only partially affecting flank marking. So both testosterone-treated hamsters, essentially summer-like hamsters, and castrates will flank mark, and they will increase their flank marking in response to vasopressin. So what this suggests is that vasopressin and some of the other mechanisms are kind of working somewhat independently. And we can see from this graph, so on the y-axis, we've got v1a receptor binding, and we can see that those long photoperiod hamsters, they have more vasopressin receptors in their brains than the long photoperiod castrates or the short photoperiods. And so what this suggests is that testosterone is increasing vasopressin in the brain, but vasopressin itself is having a similar effect on flank marking, whether there's a lot of receptors there or there isn't. And this is shown here. So those long photoperiod, those short photoperiod animals, when they're given vasopressin, they flank mark at similar rates. So here we've got flank marks on the y-axis, and we've got saline shown here. This is an odor mark cage. So hamsters will flank mark a lot when they're in an odor mark cage. If we give them a v1a receptor antagonist and we block the effect of v1a receptors, we get a reduction in flank marking, but it's not a complete blocking of the behavior. So whether they've got a lot of receptors or a few receptors, those LPs and SPs, we don't get a full ablation of the flank marking behavior in response to an odor marked cage. So what this suggests is that odor marked flank marking and vasopressin-induced flank marking are somewhat different, and they're working on somewhat independent mechanisms. So I wanted to come back to this slide here to sort of make my point again and get back up on my soapbox and say that we've gotten a long way and we've learned a lot from these sort of targeted brain region specific investigations and studies. But as these genetic tools advance, we really need to be thinking about these brain regions as interconnected networks. And having genetically modified animals is one way that we can start to piece these apart. And we need to work on the tools to do these genetic manipulations in species other than mice and rats, in species that express these behaviors that are worth studying: social communication, same-sex aggression, and social stress. So at the beginning of my talk, I promised some sort of lessons learned here. So the first thing that we learned, which is very important and has been borne out in a lot of species where CRISPR-Cas9 has been used, CRISPR-Cas9 is a very efficient method for disrupting genes and is much easier than some of the traditional methods of inducing knockouts or inducing mutations. In a brand new species, it worked very well with only a few injections. One thing that we weren't expecting and sort of tripped us up for a long time is that even though we did the injections at the single cell stage embryo, mosaicism is common and can occur. So it's probably something that should be expected and planned for, that you can't expect to do your single cell embryo injections and expect a bunch of knockouts from them or a bunch of mutants from that. That you kind of have to plan for potentially doing some breeding just to get your knockouts or to get your mutation that you want. we learned that haploinsufficiency is a very important part of this phenotype, that having one copy of the knockout allele is not the same as having two good copies or two knockout copies, and that it tends to be sort of in between. And so what this means is for future experiments, we don't want to immediately euthanize our heterozygotes, and we don't want to just save them for breeding. We want to use our heterozygotes as experimental subjects because they tend to be intermediate between our knockouts and our wild types, and can kind of give us an idea of the sort of genetic dose of having these knockout alleles. Perhaps most importantly, the results from our knockouts inform our results from our pharmacology. So even though we kind of got unexpected results, our new results from our knockout experiments can really help us sort of reinterpret some of the older pharmacological research. And now we know that we need to start thinking about v1a induced flank marking and odor-stimulated flank marking as somewhat different and somewhat independent mechanisms. And we also, as I said at both beginning and the end of this talk, is that we need to be thinking about v1a activity and brain activity from more than just a single region. We need to be thinking about brain networks. So I'm going to kind of quickly talk about some exciting future directions that we're working on. These are all very, very preliminary, and they're very much still in the works. So don't take these too far, but we're very, very excited about these future directions. So one thing that we noticed as just during normal husbandry is that our knockout hamsters seem to gain weight and specifically fat mass very quickly. And they tend to get much heavier than their wild type litter mates. So it's a little tough to see here, but both our male and female knockouts have much higher body weights than either what we call our outbred wild types, which are from the breeder, and our inline wild types, which are those that are litter mates with our knockouts. And it looks like this occurs through a steeper growth curve or a less flattened growth curve. So on the y-axis, we've got weight and fat. And in the blue, we've got our wild types as they grow from weaning to early adulthood. And you can see that the wild types, they grow quickly until they're past puberty, and then they sort of level off and gain weight slowly. Whereas our knockouts, they just continue that sort of adolescent growth curve throughout adulthood and kind of grow linearly compared to asymptotically. And this is specifically, this added weight is specifically fat weight. So this is some work that was part of a huge project from another postdoc and 3 really, really great undergraduate students. What we wanted to do is we wanted to follow our knockouts from conception to adulthood. So we did some very specific crosses. We crossed our inline, our wild type litter mate females with knockout males, knockout females with our litter mate or inline wild type males, het females with het males, and then we also did a sort of a control crossing of outbred wild type females to outbred wild type males, that should say. Outbred means that they came from the breeder. And then we observed behavior throughout their life. So we observed the mating behavior during these crosses. We observed the maternal behavior as the offspring were growing up. We observed juvenile social behavior and a novel object test. And then in adulthood, we tested them for that social defeat phenotype. So just sort of very briefly, I'll go through how we do that social defeat test. So what we do is we take a hamster and we put them into the home cage of a much larger and single housed hamster who we know is reliably aggressive and can reliably win against our smaller subjects. And then what we do is the next day we take that same smaller subject hamster and we put them into a clean cage with a different larger hamster who is sort of confined into these little plastic boxes so that they can smell them, they can hear them, they can see them, but that larger hamster can't actually get to our subject and behave aggressively. And then we measure how much time they spend on the far end of the cage, what we call sort of defeat-like behavior, versus how much time they spend near the box, which would indicate no defeat. And like I said, this is very, very preliminary. You can see there's sort of some small ends here. And what we've got here is on the y-axis, we've got that time far, which is how much time they spent on the far end of the cage away from the larger hamster. And you can see that there's some, we've got a good defeat effect here. So those black bars, the hamsters that were defeated are spending less time near they're spending more time away from the larger hamster than our no defeats, the ones that didn't get defeated the day before, and so they're good and curious and they want to interact with this larger hamster. And we found some interesting things that we found no differences in females between our knockouts and our inline wild types, but our heterozygotes were spending less time near the larger hamster, essentially showing a larger defeat phenotype. And so what this suggests is, it's not a perfect experiment because we can't get all the perfect crosses. What this suggests is that there's a combination of effects between the alleles that our subjects have, and then also the alleles or the genotype of their mother, or potentially their father as well. And so we wanted to look at that. And so we here on the y-axis, we've got that time far again, but now on the x-axis, we've got our maternal genotype. And what we can see here is that those hamsters that came from a knockout female, whether they were, in this case, they were all heterozygotes, they had a less of a defeat phenotype than our heterozygotes our offspring of heterozygote females and our offspring of wild type females. So what this suggests is that there are some maternal effects that are affecting these subjects, the grown offspring, as they interact with social information in adulthood. So I want to take the time now to thank the many, many people who were important for this project. So I noted my two co-first authors there. They're right at the top, Dr. James Walton and Dr. Kate McCann. I've bolded Elisa Norvell, who's our wonderful lab manager who made everything easier and was really sort of the through line, in addition to the PIs, throughout this whole project. She was the one who was there from conception to end. Zoe Liu did a lot of the work in just getting these embryo implants to work. It's much more difficult than it is in mice to get embryos implanted into females. Jake Vandervelden, he did a couple of the experiments independently of me as a really good replication. John Borland did a lot of work on the conception of the project. Mike Hart is our attending veterinarian. Chenglu Jin is Zoe's mentor and really pioneered these embryo implants in hamsters. Kim Human is another one of our PIs. Elliot Albers is our senior author. Dan Cox was just very, very helpful in planning the design of the guide RNA, the genotyping, and just sort of all of the methodological things that we as behaviorists didn't know that he was able to help. Our other contributors, these are all Albers Lab and Human Lab folks who just did really great work and made the lab a great place to work. The GSU Department of Animal Research resources staff made doing these experiments much easier by taking great care of our animals. Maurice Manning provided some of the chemical agents for us, specifically that v1a agonist. And then, of course, all the funding from NIH and also from Georgia State. And so now at this time, I would really love to answer any questions that people have.
Thank you very much, Jack. That was an incredible talk. I can see we've received some questions from the audience already, so I'll move straight on to the Q&A now. So your first question is, what would be the next technological advance for genetically modified hamsters?
Yeah, so I've spent a lot of time thinking about this. What would I do next? What would I do differently if I was starting this? Um, the knockout is a really great model. The knockout that we've generated is a really great model because it's just kind of easy. You breed the animals and they go. But if I had my druthers, if I had a big grant to do it, what I would really love to generate is a hamster where we've got CRE that is expressed under the same promoter as either the oxytocin receptor or the vasopressin receptor, or vasopressin or oxytocin itself. And what's really great about having CRE under the same promoter as your gene of interest is you can do a lot more things with it. With CRE, you can selectively destroy neurons that are expressing your gene of interest, or you can express something like a fluorescent indicator, fluorescent calcium indicator under your gene of interest. And so it sort of opens the toolbox a lot more. It's a much more difficult process. But given the things that we learned about using CRISPR in hamsters, and really specifically those embryo injections, getting that to work was really a phenomenal effort by Zoe Liu and Cheng-Lu Jin. Now that we know that stuff, I feel confident that if we wanted to, we could. It would be difficult, but we could generate some CRE oxytocin receptor or CRE vasopressin receptor hamsters. That would be where I would go next if I had all the money in the world.
That's fantastic. That's incredible. I'm just aware of the time, so we've got time for just one more question. And your next question is, are there any ways to time the knockout to specific developmental periods?
Yeah, so that's really important because all of our animals are born without vasopressin receptors, and so there's always this question of maybe there's some sort of compensatory mechanism that happens throughout development that is producing the phenotype that we see in adulthood. And so what would be really nice is to be able to time these deletions to specific days of age. That's where those CRE receptor or those CRE animals would be really, really useful, because you can have animals that are, they're born, they produce vasopressin receptors, but they're also producing CRE, and then you can inject them into specific brain regions or into the whole brain and tell those CRE producing cells to die or to stop producing v1a receptors, or to produce, an antisense probe. And so that would be one way to do it. Another way to do it would be, we've got some data showing that CRISPR works relatively well to knock out or knock down receptor availability in adults. But you can also do stuff with other sort of simpler viral approaches. Things like adeno-associated viruses can deliver Cas9, and they can deliver guide RNAs. Sometimes you have to do two of them. And so that would be another way, is to sort of do an injection approach where instead of injecting, like a pharmacological antagonist, you're injecting a guide RNA that's going to knock out the receptors or the gene expression in that area specifically. So yeah, that would be another place I would go next if I had all the money that I could.
That's fantastic. Thank you, Jack. Unfortunately, that is all the time we have for today. And any questions that we didn't have time to get to will be sent to Jack after the event. And as with all the other talks, the This talk will now be made available whenever you want to view for the next 30 days. So that just leaves me to say thanks again, everyone, for your time today. I hope you've enjoyed it as much as we have, and we will be back tomorrow at 8 a.m. PST with some more fantastic talks from our speakers and sponsors. Thank you and goodbye.